Welcome back, everyone. Ready for another deep dive? Today we're going to tackle a trio of cellular masterminds. You know them. You love them. Notch, Hedgehog, and white signaling pathways. We've talked about them individually before, but things get really wild when they start talking to each other, right? Especially when you think about stem cells, tissue regeneration, and even cancer.
Oh, absolutely. It's that interplay that makes it so fascinating. It's not just about what each pathway does solo, it's the whole how they regulate each other.
It's like a cellular party, but with a lot more at stake.
Exactly, high stakes.
So we're going to try and unpack some of that today. We've got a YouTube video, Cyclic AMP Signaling Part 9, and a PDF on thyroid hormone receptors to help us out. And right off the bat, the video throws us this puzzle. Why do two different receptors on heart muscle cells trigger the same cascade, but have totally opposite effects on contractions?
Yeah, that one always gets me. It's like, why two sets of brakes if they both stop the car the same way? It makes you think the one signal, one response idea is kind of too
Yeah.
simple.
Right, like there's got to be more nuance going on, and that's where the video brings up these cyclic AMP signaling microdomains, almost like different parts of the cell are getting the same message but interpreting it differently.
It's a bit like, think of a city, right? Citywide announcement goes out, but different neighborhoods react in totally different ways depending on what's going on there.
Ooh, I like that analogy. So these two heart receptors, they're both activating the same cascade, but maybe in different microdomains, and that's what leads to those opposite effects.
That's the theory. Still kind of debated, but super intriguing, especially for crosstalk between pathways like our buddies Notch, Hedgehog, and Want, right?
Now hold on, because the video throws another curveball. These things called kinase anchoring proteins, or AKAPs, and they sound like the organizers of these microdomains making sure everything's in its right spot.
You got it. AKPs are like molecular anchors, holding specific enzymes in place. One of the big ones they bind to is protein kinase A, PKA.
Wait, PKA, that's like involved in tons of cellular stuff, right? So by controlling where PKA is, these AK keys could be saying which processes get turned on or off in different parts of the cell.
You're catching on quick. That's what's so cool about AKPs. They add a layer of spatial control. So even though PKA is involved in all sorts of pathways, including Notch, Hedgehog, and Waint, it's not just running around willy-nilly.
It's like having all your tools in the right spot in your workshop.
Exactly. You wouldn't want your hammer floating around in the paint section.
No, you would not. But how do these AKPs actually, like, attach to PKA? The video mentions this amphipathic helix thing.
Yeah. It's a little technical, but think of it like a key fitting into a lock. This helix, it's got these sides that love water and sides that hate it, lets it snuggle right into PKA. And get this, every AKKP discovered so far has this helix. Pretty important, huh?
Sounds like it. So we've got these intricate microdomains organized by AKPs controlling PKA activity. Man, this is already getting complex. And we haven't even touched on gene expression yet. That's where our PDF on thyroid hormone receptors comes in.
Oh yeah, good old TRs. They're like the conductors of the gene expression orchestra. What's wild is they can act as both silencers and deactivators. Depends if the thyroid hormone T3 is around or not.
Wait, they can switch between the two, like a light switch that can turn things on A and D off.
You got it. Without T3, TRs bind to DNA and basically hit the brakes on a gene. But when T3 shows up, it binds to the TR, changes its shape, and bam, gene activation instead.
That's so cool. And I bet this dual role is super important in development, especially with all the crosstalk going on between Notch, Hedgehog, and Went.
You bet. The PDF talks about how TRs are crucial for a bunch of tissues, brain development, bone growth, the whole shebang. And just like problems with those pathways we discussed earlier, if TRs get messed up, things can go south pretty fast.
And speaking of things going wrong, the PDF even mentions this condition, resistance to thyroid hormone, or RTH. It's caused by a mutation in a specific TR gene, right?
Right. And what happens is the TR can't respond to T3 properly anymore. Switch gets stuck in the off position, and it can really mess with development.
Wow, so even tiny changes in these molecular switches can have huge consequences.
Absolutely. But TRs don't work alone. They've got this whole team of partners called cofactors.
Cofactors, what do those do?
They basically fine-tune the TR's activity. Some enhance its ability to activate or silence genes, others inhibit it, adds another layer of complexity to the whole regulatory system.
So it's like TRs have their own advisory board, helping them decide which genes to turn on or off. Fascinating stuff.
It is. And remember, we're talking about hundreds, maybe thousands of genes being regulated this way. The complexity is mind-blowing. And it makes you realize how important it is to understand how these systems work together, especially with pathways like Notch, Hedgehog, and White potentially in the mix too.
And speaking of complexity, the PDF also mentions that different TR subtypes have, like, specific jobs in different tissues, which is crazy.
Yeah.
They talk about some studies with knockout mice, right?
Yeah. By removing certain TR subtypes in mice, they figured out what each one does. One's crucial for bone growth, another for hearing development. It's amazing how specific they are.
It really is. But get this, the PDF throws in this final twist. TRs might actually be doing stuff outside the nucleus too.
Right, usually we think of them just hanging out in the nucleus, controlling genes. But this new research suggests they might have side gigs in the cytoplasm, maybe even interacting with other pathways.
Okay, that's a plot twist I did not see coming. Every time we think we've got a handle on things, there's another layer of complexity. Makes you wonder just how much we still don't know about these signaling systems.
That's what keeps things interesting, right? Always more to learn.
Definitely. So we've got these microdomains, AKAPs, the fascinating world of TRs. I feel like we're just getting warmed up. We're going to switch gears now and look at how all this plays out in the nervous system. The YouTube video, The Glycine Receptors Part 1, introduces us to, well, the glycine receptor, a key player in the spinal cord.
The glycine receptor, the main inhibitory one in the spinal cord, basically the brakes that keep our muscles from going haywire.
So without them, we'd be all twitchy and spastic.
Potentially, yeah. The video explains that glycine, which is the simplest amino acid, gets released by these inhibitory interneurons in the spinal cord. These interneurons connect to motor neurons, the ones that directly control our muscles.
Okay, so interneuron releases glycine, which then binds to glycine receptors on the motor neuron. And that's where the braking happens.
Bingo. Glycine binds, the receptor changes shape, and a channel opens up. This lets negatively charged ions, like chloride, flow into the motor neuron.
And that influx of negative charge basically makes it harder for the motor neuron to fire, right? Like puts a damper on its signal to the muscles.
Exactly. Super important for fine-tuning movement, making sure we're not all jerky and uncoordinated.
It's amazing how this tiny molecule, glycine, can have such a big impact on how smoothly we move.
Isn't it? And don't forget, the glycine receptor itself is a complex protein. It's probably regulated by all those mechanisms we talked about earlier, phosphorylation, cofactor binding, and who knows, maybe even interactions with Notch, Hedgehog, and Waint.
Oh man, the complexity just keeps unfolding. It's like we're peeling back layers of an onion, but instead of tears, we get mind-blowing insights.
I like that. And speaking of insights, we can't forget about the power of cDNA microarray analyses. That PDF on NCR talked about how researchers use those to study gene expression, right?
Right. They overexpress this dominant negative form of the core pressor NCR in mouse livers. And the results they got on cell proliferation were pretty interesting, weren't they?
Totally. Now, core pressors, like NCR, they usually act like brakes on gene expression. But in this study, when they overexpressed a messed up version of NCR, one that couldn't repress genes, they actually saw an increase in liver cell proliferation.
Wait, so taking the brakes off actually made cells divide faster? That seems counterintuitive.
It does, but it shows how complex the interplay is between signaling and gene regulation. The researchers thought maybe NCR normally keeps genes that promote cell division in check, so by messing with NCR, they accidentally stepped on the gas pedal instead of the brakes.
Whoops. Talk about unintended consequences.
Right. But this kind of unexpected finding opens up new ways to think about how core pressors like NCR might be involved in diseases like cancer, where out-of-control cell division is the big problem.
Okay, so we've gone from microdomains in heart cells to gene regulation in the liver, and now to the workings of the glycine receptor in the spinal cord. It's amazing how these seemingly unrelated things are all connected by cell signaling and gene regulation.
It really shows how unified biology is, how the same principles are at work across all these different cell types and tissues.
It's beautiful, really, when you think about it. But before we get lost in the beauty, let's try and connect all these details back to those 3 developmental pathways we started with: Notch, Hedgehog, and Went.
Ah, yes. Our 3 maestros. Now, our sources didn't go too deep into how they interact with TRs, NCRR, or the glycine receptor specifically.
It's like they're lurking in the background, right? Subtly influencing the whole show.
Exactly. What's so cool about Notch, Hedgehog, and Went is that they're involved in pretty much every aspect of development, from the very first stages of an embryo to keeping adult tissues running smoothly. And they often work together. Their signals kind of converging to make those crucial decisions about what a cell will become.
So it's not just one pathway dictating what a cell does. It's this whole complex conversation between multiple signals.
You got it. And the outcome of that conversation can change depending on the context, the cell type, the developmental stage, other signals floating around.
Like a recipe where even a tiny change in ingredients can lead to a completely different dish.
Delicious analogy. Now, imagine these pathways interacting with all the systems we've talked about today. TRs controlling genes, NCOR tweaking that control, the glycine receptor fine-tuning neurons. The possibilities are pretty mind-boggling, right?
I'm starting to see this grand symphony now. All these pathways of molecules playing their parts, interacting with each other, responding to cues from the environment. It's incredible.
It is, and remember, this is just a snapshot of what we know so far. There's so much more to uncover about how these pathways connect, how they're regulated, and how things go wrong in disease.
Well, on that note, I think it's time to shift gears again. We've laid the groundwork with microdomains, AKAPs, and those amazing TRs, but now let's see how these principles play out in a whole new context. The nervous system.
Sounds good to me. Always up for a new adventure in the cellular landscape. Makes you wonder if we'll ever fully map it all out, you know? Like, it's this massive network of switches and signals, each one with its own little quirks, and now we're trying to figure out how they all fit together in this big old symphony of development and disease.
It's like a giant jigsaw puzzle, right?
Right.
We found some of the corner pieces, maybe a few
Right.
from the edges, but the middle's still a big mystery.
Exactly. But that's what makes it so exciting, isn't it? Each new piece we find opens up even more questions.
Definitely keeps things interesting. And when it comes to Notch, Hedgehog, and Went, well, it's like we've got three puzzles going on at the same time, and sometimes the pieces overlap.
Oh, absolutely. They're not just doing their own thing. They're constantly interacting, influencing each other, sometimes even fighting for control.
It's like trying to follow three conversations at once.
It can be. But thankfully, researchers have come up with some pretty clever ways to untangle it all. Like they can use inhibitors to block one pathway at a time and see what happens to the others.
Ah, that's smart. Like muting one instrument in an orchestra so you can hear how the others change their tune. So what have they figured out so far about how Notch, Hedgehog, and Went actually, talk to each other?
Well, it's not a simple answer, that's for sure. Sometimes they cooperate, like they're all on the same page, amplifying each other's signals to get something done. But other times they're totally at odds, like a tug of war, you know?
The cellular tug of war.
Yeah.
I like it. Makes sense, though, right? It'd be too simplistic if they are always friends or always enemies.
Right. It's all about context. And this kind of push and pull becomes really important in stem cell niches, those little microenvironments where stem cells hang out and get their instructions.
Ah, stem cells, the blank slates of the body, right?
Mm.
They can become any cell type. So I bet the interplay between Notch, Hedgehog, and Went is crucial for telling them what to do.
Crucial is an understatement. In lots of tissues, these pathways work together to maintain the balance between stem cell self-renewal, where they just make more stem cells, and differentiation, where they specialize into specific cell types.
So it's like keep enough stem cells around for the future, but also making sure we've got the right cells for what we need right now, Adele.
Yeah, exactly. And that balance, it's got to be perfect, otherwise things start to go wrong. Like if the signals get messed up, that balance can tip, and that's where things like cancer can come in.
Oh, right, because cancer is basically cells growing out of control, right? So if those pathways are sending the wrong messages, telling cells to keep dividing when they shouldn't.
That's one way it can happen. It's like the communication breaks down and the cells go rogue. And that's why understanding this crosstalk, not just in normal development, but also in disease, is so important.
Makes sense. But man, it sounds complicated.
It is, but there are some really bright people working on it, and they're coming up with some cool new approaches. Like some researchers are looking into targeting multiple pathways at the same time instead of just one.
Oh, interesting. So instead of trying to silence just one instrument, we'd be adjusting the whole orchestra.
That's a great way to put it. The idea is that by hitting multiple targets at once, we might be able to overcome the resistance that sometimes happens with single target therapies. Like, if you block one pathway, the cancer cells might find a way to use another one to keep growing.
Ah, so we got to be smarter than the cancer cells.
Exactly. Got to outsmart them. And that means understanding the whole network, all the connections and feedback loops. Is like if we can crack the code, then we can start to really control the system.
I'm really starting to grasp the scope of this now. It's not just about individual molecules or pathways. It's about how they all work together in this incredibly complex and dynamic network.
You got it. And that's why research in this area is so exciting. Every new discovery brings us closer to understanding the big picture.
Okay, so we've talked about how Notch, Hedgehog, and Want can kind of go rogue and contribute to things like cancer, but they also play a huge role in tissue regeneration, right? Like those animals that can regrow limbs, that's got to be thanks to those pathways, right?
Yeah. Oh, absolutely. Those regenerative superstars, they've figured out how to use these pathways to their advantage. Like, take the salamander, right? Loses a limb, no big deal, grows a new one.
It's like something out of a comic book. How do they do that?
Well, when the limb gets lost, the cells at the wound site, they basically revert back to a more primitive state, kind of like stem cells. It's called de-differentiation.
And they're like rewinding their developmental clock. That's amazing.
It is. And that's when Notch, Hedgehog, and Want really kick into gear. They orchestrate this whole cascade of events. Cells start dividing, migrating, differentiating into the right cell types, and bam, new limb.
It's like watching a construction crew rebuild a building from scratch.
But how do the cells know what to do? Like, how do they make sure the new limb is the right shape and size and has all the right parts?
That's the million-dollar question. We don't have all the answers yet, but it seems like those pathways are responding to cues within the tissue, like a blueprint that tells each cell where to go and what to become.
It's like they have their own internal GPS system.
Right. It's incredible. So why can't we generate limbs like that?
The age-old question. Sadly, our regenerative abilities aren't quite as impressive as some other creatures.
So we're just stuck with our limitations.
Well, not necessarily. We do have some regenerative capacity, just not to the same extent. And researchers are trying to figure out how to unlock that potential, how to boost our natural repair mechanisms.
That's good to hear. So even if we can't regrow entire limbs, maybe we can still improve our body's ability to heal.
Exactly. And guess what? Those same pathways, Notch, Hedgehog, and Want, they're key players in the types of regeneration we can do, like wound healing, liver regeneration, even repairing damaged heart muscle.
So they're not just superstars and salamanders, they're working hard in our bodies too.
All the time. And by understanding how they operate in these contexts, maybe we can figure out how to give them a little boost, help them work even better.
That's awesome. But I imagine it's not easy, right? Messing with these pathways, it could have unintended consequences.
Oh, definitely. It's a delicate balance for sure. But researchers are working on ways to deliver those signals more precisely to the right tissues at the right time.
Like fine-tuning the orchestra, making sure all the instruments are playing in harmony.
Exactly. It's a challenge, but the potential rewards are huge. Imagine being able to help people heal faster, regenerate tissues that were thought to be lost forever. It's mind-blowing.
It really is. This whole deep dive has been mind-blowing. We've gone from tiny microdomains to the grand scheme of regeneration. And I feel like we're just scratching the surface.
We are. But that's the beauty of science, isn't it? There's always more to learn, more to explore.
Well, on that note, I think we should take a break from the mind-blowing stuff for a moment and give our listeners a chance to catch their breath. We'll be back soon to wrap things up and leave you with some final thoughts to ponder.
Yeah?
It's kind of scary, right? These pathways are doing all this good stuff, building our bodies, keeping us healthy, but then they can also go haywire and cause all sorts of problems.
Yeah, it's like they have this dark side. But I guess that's true for a lot of things in biology. Too much of a good thing can be bad, right?
Right. It's all about balance. And that makes me wonder about all those therapies we talked about, the drugs, the immunotherapy. Are there any other approaches researchers are looking at?
Oh, tons. One area that's really hot right now is the tumor microenvironment. That's all the stuff surrounding the tumor, the cells, the blood vessels, all of it. It can really impact how the tumor grows and spreads.
Oh, yeah. I remember hearing about that. It's like the neighborhood the tumor lives in can either help it thrive or keep it in check.
Exactly. And guess what? Notch, Hedgehog, and Want, they can actually influence that neighborhood. They can mess with the immune cells, the blood vessels, all sorts of things.
So they're not just making the cancer cells go crazy, they're also remodeling the whole area to make it more cancer-friendly.
Yep. Pretty sneaky, huh? But the cool thing is, if we can understand how they're doing that, maybe we can find ways to disrupt it. Like, make the microenvironment less hospitable to the tumor, maybe even boost the immune system so it can attack the cancer cells more effectively.
That's brilliant. It's like cutting off the enemy's supplies A&D, sending in reinforcements at the same time.
Exactly. And researchers are even thinking about combining these strategies with chemo and radiation, hit the cancer from all sides.
A multi-pronged attack. I like it. Makes sense, though, right? Cancer is such a complex disease, it probably takes a combination of approaches to really tackle it.
You're getting it. It's like a puzzle, and we need all the pieces to see the full picture.
Well, I think this deep dive has definitely given us a lot of pieces to think about, from those tiny microdomains to the grand scheme of regeneration, and even the fight against cancer.
And don't forget those cellular DJs, Notch, Hedgehog, and Went, spinning their signals and remixing the whole system.
They're the life of the cellular party. But in all seriousness, it's been an incredible journey exploring all this with you.
Likewise. Always a pleasure to nerd out about this stuff.
And I hope our listeners have enjoyed it too, even if their heads are spinning a bit now. It's amazing to think that all this is happening inside us right now, at this very moment.
Right. It's mind-blowing, but it's also kind of beautiful, don't you think? All these tiny molecules working together to create this incredible symphony of life.
Absolutely.
Yeah.
It makes you appreciate the complexity and the elegance of biology. And it gives me hope that as we learn more, we can unlock even more secrets and find new ways to heal, to regenerate, to thrive.
Couldn't have said it better myself. Keep exploring, everyone. Keep asking questions. Keep diving deep.
And until next time, keep those cellular DJs spinning. Thanks for joining us on the Deep Dive.